Hi everyone, I am Dr.J.BHUVANA, Assistant Professor of Chemistry, A.D.M. College for Women, Autonomous, Nagapattinam. Today, we are going to discuss about mechanism of drug action. This module belongs to second BS Chemistry in Pharmaceutical Chemistry paper. So, what is meant by mechanism of drug action? The action of drug is the process by which the drug brings about a change in some pre-existing physiological function or biochemical process of the living organism. The effects produced by a drug can be determined and expressed only in terms of an alteration of some known function or process that maintains the existence of the organism. Any alteration that is brought about by the action of a drug may be one that either returns a function or process to normal operating levels or changes a function or process in a direction way from the normal level. A particular effect observed as a change in definite physiological process may be brought about in several ways. The action may be of the drug may be specific, that is, it aimed directly as the agent responsible for the disease. Okay, so it may be specific, that means the agent that responsible for the disease is aimed directly, or it may be non-specific, that means improving a system of the disease such as fever without getting to the basis of the disorder that can be controlled. If the drug agent is non-specific, means it improves the system of the symptom of the disease, such as fever we got for any reason. The effect of many drugs or their actions can be characterized and identified only in terms of the knowledge of their physiological function and the processes. The part of the body, namely organ, tissue or cell, where a drug acts to initiate the chain of events leading to an effect is known as site of action of the drug. So, what is the definition for site of action of the drug? When a part of the body, namely organ, tissue or cell, where the drug acts to initiate the chain of events, that means the curing events leading to an effect is called site of action of the drug. For example, the site of action of quinine, a drug used for malaria, is the plasmodium. The plasmodium enters into the cell invading the host, whereas in non-malarial fever, the drugs act at the discrete areas of the brain involved in the regulation of the body temperature. So, to explain the site of action of drug, we explained using morphine. Next, the site of action and the effect of drug both are mutually dependent on each other. So, the site of action and the effect of drugs are mutually dependent. Why? For example, the effects of drugs on the pupil of the eye, the muscles of the iris control the size of the pupillary opening in response to light. That is, in bright light, the opening is made smaller, and in dim light, it is larger. Some drugs, like atropine, when applied to the external surface of the eyeball, cause the pupil become larger than it would be normally under the same light conditions. The ophthalmologists use atropine or similar agents to enlarge the pupil, which helps them to examine the eye. On the other hand, solutions of the morphine, when applied externally to the eyeball, there is no change in the size of the pupil, but when morphine is given internally, it produced marked constriction. So, when the morphine is applied externally, there is no change. When morphine is given internally, it produces constriction. In fact, this effect is so characteristic that the presence of small or pinpoint pupils lead to the perception that an individual has taken morphine. The site of the morphine is within the brain. Thus, when morphine is administered internally, it reaches this site of action within the central nervous system. Generalizingly, about the site of drug action, it is easy and conclude that it can be derived by process of elimination, but by precise determination of the specific site and the mechanism of the drug is very difficult. And to determine the site at which a drug acts to produce a specific effect requires detailed experimental studies. For example, it is easier to show that the iris of the eye is not the site at which morphine acts to constrict the pupil, but to determine the site where morphine acts require extensive investigations to find out in which manner individual areas of the brain influence physiological functions. Even after determining the site of action, so if the drug enters, even after determining the site of action of a drug to a certain tissue of organ, it does not give sufficient idea about the effects of different drugs, since different drugs producing same or different effects may have different site of actions within the same tissue or organ. For example, camphine, histamine, nitrites like nitroglycerin, these all act on the muscle of the blood vessels to increase their diameter, whereas cocaine produces constriction. So, it is necessary to study the separate components of the tissue or organ, and it has to be determined that which of them is the specialized function element on which the drug acts. These studies help to show that the site of action is a particular kind of cell within an organ, and that the drug acts either at the surface of the cell or within the cell itself, or the site of action can be extracellular. That is, the site of action can be outside the cell. The site of action of each drug is very specific in nature. So, next term is mechanism of action. How the drug acts upon your body. When the site of action of the drug is determined. So, we determined now the site of action. How the mechanism is take place. It can be either at the surface of the cell or extracellular or intracellular. The next step is to find out how the drug acts. That is, the means by which a drug in the immediate vicinity of its site of action initiates the chain of events and which can be measured as an effect is known as mechanism of action. That means the drug, how much it reacts, this is called as mechanism of action. The mechanism of action of most drugs involves a chemical interaction between the drug and a functionally important component of the living system. So, how the mechanism of action takes place? It can take place by three ways. So, what are they? A drug may act extracellularly on the cell surface inside the cell. What are the examples for this type? Extracellularly means, example is osmotic diuretics, plasma expanders. On the cell surface means, example of the drug is penicillin, insulin, etc. Inside the cell means, steroid hormones are the best examples for the drug action inside the cell. So, let us discuss one by one. So, first one is action at extracellular site. The drug action occurs outside the cell. One of the simplest example is the neutralization of excessive gastric acid by antacid drugs. So, when we eat large number of amount of food, we eat what type of pills? Digest. It is act as a antacid drugs. The antacid drugs neutralize the excessive secretion of gastric acid in our stomach. In this reaction, a base such as sodium bicarbonate reacts chemically with the hydrochloric acid of the stomach and neutralize the acid. Okay, and neutralize the acid by formation of new products. What are the products are formed? Salt, water and carbon dioxide. So, this one is the first one. Another example for this type is specific antidote. That means we take calcium disodium edetate, which is used for the treatment of lead poisoning. This drugs removes lead that is free in blood and tissue by a reaction in which the calcium of the drug is displaced by the lead. This drug has much more affinity for lead than calcium. Hence, this drug forms an inert complex with lead and to excreted it from the body as the soluble lead edetate complex. The study of these examples make it clear that the action of drug at the extracellular sites is mainly due to chemical reaction only. So, the chemical reactions are very much responsible for the action of extracellular. These chemical reactions can be easily explained as drug involved and the other reactants are specifically identifiable. The mechanism of action of drug at extracellular sites can be readily explained since the drug involved and the functionally important molecule of the living system. Second one, what is second one type? Action at cellular sites. Most function of the living organisms do not take place extracellularly, but they take place cellularly. That is, most drugs combined with some molecular structure on the surface or within the cell. This molecular structure is called as receptor. The combination of the drug with the receptor results in a molecular change in the receptor like altered configuration or charge distribution and thus triggers a chain of events leading to a response. So, what is meant by drug receptors and the biological responses we have to know and discuss. So, receptors are actually macromolecules present in tissues that combine chemically with the drug. Most drugs have considerable degree of selectivity or specificity in their actions and hence The receptors with which they interact must be equally unique. That means the drug acts at some sites to produce its characteristic biological effects, whereas its presence at other cells or tissues or organs have no biological response. The receptors are highly specialized macromolecules which recognize the drugs which are highly specific and selective in their action and trigger a chain of events which ultimately result in biological response. Hence, it suggests that there are some specific physiochemical properties of the biological molecules at the site where the responses are produced by drug interaction, and these specific properties are missing at other sites. There is also a definite relationship between the chemical structure of the drug and the biological effect it produces at a particular site. Even a very slight modification in the chemical structure of the drug may yield a compound which is no longer effective at the site as the parent compound. Sometimes it is found that there are changes in the effect produced at the site, although there is no change in the chemical formula of the drug. But there are only changes in the way a few atoms are arranged in the space or in the three-dimensional structure when the compounds are stereoisomers. A drug can produce an effect only it fits with its receptor as a special key, fits a well-designed lock. The drug receptor interaction can be explained through a specific example for your easiness. The end plate region of a skeletal muscle fiber, which contains a large number of receptors which have a high affinity for the transmitter acetylcholine. Each of these receptors, known as nicotinic receptors, are part of a postsynaptic membrane that controls the inward movement of sodium ions. At rest, the postsynaptic membrane is relatively impermeable to sodium, and stimulation of the nerve leading to the muscle results in the release of acetylcholine from the nerve fiber in the region of the end plate. The acetylcholine combines with the receptors and changes them so that the channels are opened and the sodium flows inward. The most acetylcholine that is in the end plate region, the more the receptors that are occupied and the more channels are opened. When a large number of channels are opened, then after some time they reach a critical number when sodium enters rapidly and disturbs the ionic balance of the membrane and it depolarizes. An action potential develops. Due to depolarization, there is a release of calcium from intracellular binding sites. This calcium then interacts with the contractile proteins, resulting in shortening of the muscle cell. The nitrogen atom present in acetylcholine acquires a N or quaternary ammonium compound by donating its unshared pair of electrons to the carbon atom, and this nitrogen atom forms an ionic bond with the negative charge of the receptor. This electrostatic attraction is sufficient to draw acetylcholine close to the receptor and the stability of the bond is attained by van der Waals attraction produced by close fitting of two of the methyl groups into the cavity in which the charged site of the receptor is embedded. Another hydrogen bond with oxygen draws the other end of the molecule close to the receptor and further increases the stability of entire acetylcholine receptor combined. The sequence of events by which acetylcholine brings about muscle contraction through receptors. The same way drugs can be designed to interact. A chemical with a molecular structure very similar to that of acetylcholine might be attracted to the receptor in place of acetylcholine and changes the receptor's molecular orientation in a manner which is similar to that of acetylcholine, and this initiates the chain of events that causes muscle contraction. On the other hand, if a chemical is less similar acetylcholine, it may interact with the receptor, but it is unable to induce the exact molecular change necessary to allow the inflow of sodium. This chemical does not cause contraction at the same time, but because it occupies receptor site and inhibits the interaction of acetylcholine with its receptor. What is the chemistry of drug receptor binding? So we have to know the drug receptor binding. Many biological receptors are composed of proteins, nucleic acids or other macromolecules. These macromolecules combine with a drug in a number of ways, and the forces by which a receptor combines with the drugs are fairly strong and long lasting, so that the sequence of biological response could be completed. These forces are known as chemical bonds, and a number of different types of bonds participate in the formation of the initial drug receptor complex. So what are the bonds involved in the drug receptor bonding? First one is covalent bond. The bond formed by a sharing a pair of electrons is called covalent bond. It has a bond energy of 100 kilocalories per mole, and therefore it is strong and stable and is responsible for the stability of most organic molecules. Since covalent bonds are stable at physiological temperature, it results in the formation of a long-lasting complex, and it can be broken only if sufficient energy is added or by a catalytic agent such as enzyme. Covalent bond formation is found in anti-neoplastics of antibiotics, where long-lasting inhibition of cell replication is required. If both the shared electrons that form the covalent bond are derived from only one of the two atoms, the bond is called coordinate covalent bond. The atom contributing the electron pair is called the donor atom, and in biological system, the donor atom is usually nitrogen, oxygen or sulfur. For example, ammonium ion is formed by the reaction of ammonia and hydrogen ion. The atoms other than hydrogen atom usually that share the donated electron pairs or cations such as sodium, potassium, copper, zinc, magnesium, etc. This coordinate covalent bond formation is important in the ionization of drugs and in certain interactions with the receptors and in chelation reactions. The chelation reactions are between a chelating agent and a metal cation. Chelation reactions are often important in binding substrate to their enzymes, and they form the basis for a number of pharmacological and toxicological effects produced by the administration of metal containing or metal chelating drugs. Next bond is co-hydrogen bond. The hydrogen atom is strongly electropositive in nature when it lies between two atoms having electronegativities like oxygen, nitrogen or fluorine, shows a unique property to form a bond and bridge between them, holding one by covalent bond and the other by purely electrostatic forces. It is known as hydrogen bond. What are the mechanism of different types of drug action? How the drug acts upon our body? Various drugs produced their effect by different processes such as by depression, by stimulation, as replacement, as anti-infective agent, as anti-metabolite antagonist, by irritation, by modification of immune system. So the drug produced different process by 7 processes. They are by depression, by stimulation, as replacement, as anti-infective agent, as anti-metabolite antagonist, by irritation, by modification of immune system. So let us discuss one by one. So what is the first type? Depression. The action of these drugs is to bring depression of some special cells and decrease their activity. So some example for this type is barbiturates, which depress the central nervous system and used as sedatives, hypnotics and analgesics to reduce pain and also to control convulsions in epilepsy. Next one is by stimulation. These drugs stimulate the activity of specialized cells and thus increase the activity. Excessive stimulation produces changes in the protoplasm of the cell, which may lead to the depression of certain portions. Thus, these drugs stimulate certain portion and depress other systems. For example, morphine, which stimulates the nerve situated at medulla oblongata but depresses the vomiting and the cough centers. Next one is replacement. These drugs are used as replacement when the production of endogenous substances is reduced. These drugs are of immense importance in the treatment of disease or hormonal deficiencies. Example, insulin used in diabetes mellitus. Next type is anti-infective agents. These drugs are used for prevention and treatment of infection. They either stop the growth of the microorganism or immediately destroy them. Examples of such drugs are antibiotics like penicillin, tetracycline, etc. Antibiotics are chemical substances produced by living cells only, which are capable of inhibiting or destroying other microorganisms. Next type is as anti-metabolite antagonist. These drugs are bacteriostatic in action and act as metabolite. That is, it interferes with the metabolism of the bacterial cells and thus inhibit the growth. Example for this metabolite is sulfur drugs. For the sulfur drugs, we take sulfonamide as example. This sulfonamide drug structurally resembles para-aminobenzoic acid, which is a normal cellular constituent or metabolite of the bacteria. Bacteria takes up sulfonamide due to their structural similarity to that of para-aminobenzoic acid. The sulfonamide cannot perform the essential action of para-aminobenzoic acid metabolite, and hence the growth and the production of bacteria ceases. That means it will stop the growth. Next one is by irritation. So it is the term which indicates that a drug acts by affecting the growth, nutrition and the morphology of living tissue cells. The cellular changes produced by irritation are of precipitation of protein or corrosive effect, that means damage to the tissues or dehydration or enzyme neutralization or damage to the cell wall. Heavy metals like mercury, silver are used as irritants. Fifth type is modification of immune status. When the affected organisms enter the body, that means the infected organism enter the body, they start manufacturing toxic antigens into the body. The human body biological deficiencies start producing antibiotics which destroy these antigens. Immunological agents produce active or passive immunity which are used to prevent or modify certain infectious diseases. These agents are known as vaccines, and when these are administered in the body, it simulates the body for manufacturing antibiotics in defense against the disease. Some infectious diseases like cholera, smallpox, typhoid, diphtheria, tetanus can be prevented by this way. So, let us came to an end. We discuss about mechanism of drug action, that is in your pharmaceutical chemistry paper. Thank you.